An Agrichnial feeding strategy for deep marine Paleogene Ophiomorpha group trace fossils. In the article as a vertical to horizontal burrow displaying a distinct lining comprised of muddy pellets that are typically created by invertebrates such as crustaceans. They first appeared in the Permian and were found to be constructed in shallow water. The direction of the Ophiomorpha, vertical or horizontal, is said to be indicative of the weather conditions when they were formed, either good weather versus storm weather. They became common in the Cretaceous and deep marine environments. These specimens have been found in northern Spain. The question being tested is, an Agrichnial feeding strategy for deep marine Ophiomorpha? Some modern-day species of shrimp who feed agrichnially construct Ophiomorpha-like burrows. The Y-shaped burrows also connect to create a partial or complete hexagonal pattern. Other trace fossils, especially those of Nereides ichnofaces, show similarly shaped complex burrowing patterns and have been said to show that the species that created them use them for agrichnial feeding as well. All of these examples suggest that the organisms that created these Ophiomorpha use the same agrichnial feeding strategy. The burrows are organized and connected through networks. This pattern supports the hypothesis that Ophiomorpha were used as an agrichnial feeding strategy for their inhabitants. It is presumed that the burrows were open to the seafloor, causing irrigation of the burrow and consequently the flow of organic materials to enter the burrow. The discovery of deep marine specimens in northern Spain show that these Ophiomorpha types of burrows, which originated in shallow waters in the Permian, progressed to deep marine environments in late Triassic. Because of this progression to deep marine environments, the animals that create Ophiomorpha must have also progressed in some way through adaptations in order to survive in such a different environment. The construction of the Ophiomorpha burrows examined in this study do suggest that Ophiomorpha support agrichnial feeding. A change in the currents bringing food to these deep marine Ophiomorpha could be an effect of global change and could have also affected the development of the burrows in such deep marine conditions.
The next few slides are going to focus on trace fossils of vertebrae, specifically the crocodilomorph eggs and eggshells from the Adamantina Formation of the Barrow Group in Upper Cretaceous of Brazil. Oliveira and his colleagues discussed the surprisingly well-preserved eggs and eggshells of the fossil crocodilomorph, and based on the various characteristics such as the shell size, shape, shell width, they identified these eggs as a new ootaxon, Barothilus fragilis. So, the Barothilus fragilis is the first oo species described from South American deposits. It was found in the Adamantina Formation of the Barrow Group in Brazil, and it was dated to be from the Cretaceous period. These maps on the side show where the egg clutches were found in Brazil and their locality. This is the first crocodiloid ootaxon with several egg clutches recovered, but unfortunately none of the eggs that were collected are complete. So the question is, what is the paleobiological and functional significance of this new ootaxon? The shell fragments were studied using light and polarizing microscopy, or shortly PLM, and scanning electron microscopy, or SEM. Which allowed for the evaluation of morphological variation in different portions of the same egg. The system of levels of confidence was used in order to establish a relationship between the oological material and the egg-laying taxa, and this system was proposed by Grellet and Tinner in their articles of 2005 and 2006. 15 eggshells were measured in order to determine the shell thickness. The PLM system and the stereoscopic zoom was used for this. They compared their findings to those previously as shown in this table. So, through their analysis, they determined that the shape, shell thickness, and size of the new eggs are crocodiloid. The Barothilus fragilis ootaxon is characterized by its elongated and elliptical eggs with their blunt ends. The length and diameter ratio of the eggs are 1 to 0.55. Its shell thickness ranges from 0.15 to 0.25 meters. What's fascinating is that they determined that the shell thickness and the lack of signs of extrinsic degradation in Barothilus suggests that the embryos didn't need the extrinsic degradation to facilitate the hatching process. And if there are signs of extrinsic degradation, there's a high chance that this was an instance of premature hatching. So in conclusion, these eggs and eggshells are important for understanding eggshell morphology, and these finds have increased the diversity of fossil eggs from Brazil. This paper has also established the problems with size and shape identification of fossil crocodilian because they can be somewhat unreliable due to the fact that size and shape can be affected by preservation. So shell thickness can be used as a more reliable approach.
Now we will go back to looking at marine life. This article explores microfossils and is entitled The First Trace Fossil Evidence of Bone-Eating Worms in Whale Carcasses. In order for you to better understand the conclusions brought forth by the researchers of this article, background information is much needed. When whales and other marine animals die, their deceased bodies or carcasses are food for numerous life forms. Paleontologists looking at the Neogene basin among the Pliocene rocks in southeastern Spain found round microfossil borings, which are similar to holes or little caves, in the cranium of a deceased baleen whale. This picture shows the geological location where these fossils were found and the different strata, which are color-coded in the key. So the question at stake, or the question that the scientists of this article want to answer, is what organism is responsible for the tubular borings, which are the trace fossil evidence, in the cranium fragments of this whale carcass? So when they closely looked at the actual borings, they found that they were unbranched, they were cylinder-shaped, and they were perpendicular to the whale bone surface. So then they were able to identify these worm borings, or the trace fossil evidence, as Trypanites ionaceae. And on the next slide is a high-resolution image depicting these borings. Further analysis of the worm borings led researchers to conclude that it could possibly be the worm species Osedax. So they compared the female Osedax anatomy and the boring dimensions and found that it exhibited many similarities. This table shows the comparison. They looked at the anatomy of the Osedax female's root, body length, and trunk, and found that it lines up nicely with the boring's dimensions. Therefore, researchers concluded with a high certainty that the borings Trypanites ionaceae could indeed be the consequence of the siboglynnid polyhedrid Osedax's behavior. The larger females may have bored into bone with the help of Oceanospirales bacteria, which formed a symbiotic relationship. Although researchers have acceptable evidence to support their claims, additional support is mandatory to strengthen their conclusions. So further analysis of the specific bioerosion of Osedax species is beneficial for an increased certainty.
And now we're going to transition from talking about whales to talking about plants. So the paper I'm examining for this section explores fossils of conifer tree roots from the Quaternary on New Zealand's North Island and in Georgia in the United States. And according to my paper, trace fossils for plants are actually quite common and have been identified since they started identifying fossils. However, they are less well classified and understood compared to vertebrate trace fossils. The main questions, there's two main questions posed by my paper. The first of these is, are the fossils we're finding body fossils or trace fossils? And the second question is, what do these trace fossils, if they are indeed trace fossils, reveal about the paleo environment? To answer this question, the authors of my paper first looked at the Karikari and Apori peninsulas on northern New Zealand. Both these peninsulas were once heavily forested with giant kauri pines 40,000 to 2,000 years ago. However, because of deforestation, the area's dunes are now subject to erosion. This erosion, coupled with a road they've been digging in the area, has revealed 3 different types of plant trace fossils. The first of these are molting ichnofabric plantolites, the second branching thalassinoids, and the third are large-scale pipe-like structures that have not been solidly identified. So here are some pictures of the site on your, I guess this would be your left. You can see a picture of the dunes, and on your right you can see some of the trace fossils. Here are some more pictures of the trace fossils. You can see the dark represents what remains of the roots, while the light-colored soil represents the sand around the trees. Interestingly, around these plant trace fossils, scientists have also found a number of invertebrate trace fossils too. Strikingly similar to New Zealand, scientists in the United States have, at Sapelo Island, found very similar trace fossils. Now, Sapelo Island is a barrier island that used to represent the extent of the Pleistocene coastline, and is perhaps the best studied barrier island ecosystem. In analyzing the trace fossils at Sapelo Island, scientists have discovered that the Quaternary forests of the coastline were more similar to modern barrier island ecosystems than they are to modern coastline ecosystems. So based on the analysis of these trace fossils, scientists have discovered that the trace fossils from late Pleistocene New Zealand are in fact very, very similar to similar root complex structures found in early Holocene Georgia. These fossils reveal numerous instances of microbial and cicada activity. Additionally, unidentified trace fossils found in the vicinity of these root trace fossils suggest a wide variety of other invertebrate life was present as well. So these finds are important because they add a new dimension to the study of Quaternary paleoenvironments. They also explicate the relationship between burrowing terrestrial invertebrates and piney root systems, which has bearings even today. To finally summarize, trace fossils are useful for discovering all sorts of things about the ancient world, from the habitats of burrowing worms to life in Quaternary forests. Although they may not be as exciting as body fossils, trace fossils are valuable in understanding our Earth's past.